So it's time for the panel discussion session, apparently the most engaging session of this conference. Um, so we will have to play by the rules because we have five, presenters, and 5 plus 2 is the allocated time this session. Seven presenters. Okay, we have seven presenters. We have seven presenters, so we will have 5 minutes each, so that we'll still have 10 minutes for general questions. So the way I want to work this session out is each presenter will have at most 5 minutes to talk, and our timekeeper, as soon as he says zero, which means he'll have to stop, because we just don't have extra time. Unless you have a pressing question to ask after the 5 minute presentation, we'll keep all the questions for the later half, so that we can kind of efficiently organize the deliberations here. So the way I want to call the presenters, so the first one will be Dr. Binay Panda, because he will talk on open platforms. Then I will ask Varun, Dr. Varun. Yeah. So since I think your will be experimentation, right? So I want experimentalists to go first. Then Varun will be followed by Saket. After Saket, I would want Nitya to come in. And then after Nitya, it will be Mile. And after that, last one would be Paul. Yeah. So your presentations. Okay. Um, yeah. So Binay, you please get started, and uh, we will try to keep to the time. Otherwise, last year we had a extended session, but I guess we'll try to keep the time under control.
Any slides, so that makes it easier to talk. Okay, so what I'm going to talk about is when I was asked to be in the panel and asked to give the topic that I want to talk, I wanted to be generic because I thought the panel discussion is meant to enhance our ability as a community to come together, not to tell about I need this, you need this. No, panel is for everybody, for everyone in the country. What we need to do to put computational biology community to the next platform. And I'm going to talk about open data. The reason I'm going to talk about open data, what needs to happen where I see it, and please feel free to refute it, is why do we need open data? What is the need of an open data for a computational biology community or a bioinformatics community? Why do we need it? Do we have it? And the answer is obviously none. And how do we build it? And how do we sustain it? Building is easier in our country than sustaining it. So how do we build, sustain an open data community, which is going to be a fuel for students and the PIs to use it to build tools, to benchmark, to use that as a ground truth. So you heard in the last couple of days, almost every single presenter talked about using data from a database or multiple databases. You heard about HapMap. I'm sure that all of you probably know, the old guys will know about HapMap. GTEx, genome in a bottle, the sequencing data which is deposited in the database in the beginning, and Genomics England data. All these are the databases that we use in a daily basis to not just develop our tools, to benchmark our tools, but to make sure that we have a gold standard. Now, this is great. Then why do we need to build one in India? You may ask. The Americans, the Europeans, the Japanese are building it. We are using it. The scare came in, many of you may remember, in 2006, if I remember the year right. There was a scare that SRA is going to be closed. You guys know. I mean, we panicked. We said that's kind of a bread and butter we download every day is going to be closed. And there was a huge hue and cry, of course in the US, but also around the globe. But don't you think it's a little selfish on our part to rely on a database that we didn't fund? We didn't fund to start, we didn't fund to sustain, but we still demand that the database should be open. All of you download data from SRA. Have you thought about it? Who pays to maintain that? The exponential growth. I mean, I think last time I checked, it was close to 92 petabytes, right? So we must build databases on our own and help sustain the databases out. Now, very quickly, I'll tell you why do I need to build our own. You heard the last talk from Devashish, the population genetics. Now, the allele frequency is very population specific. If you are looking for a recessive disorder which is only in one endogamous population in India, what do you compare with? You can't compare with Ashkenazi Jews population to Caucasian population. If I am looking at the germline mutation of a particular solid tumor, I need to compare with another population which is close genotypically with the population that I'm studying. Therefore, building databases and building data on Indian population is critical. Remember, here the term India is very loosely used. I understand that there are multiple different genotype groups in India, 4 or 6 depending on who you talk to. Now, we need to build databases, we need to fund databases, we need to sustain databases for that purpose. And that doesn't exist today. If you are in rare diseases community, there are many rare diseases that are only found here because we have a large endogamous population, we have a large consanguinity in our country. And of course, it's also present in Middle East, but the diseases could be different, the manifestations could be different. So the time has come where we need to build, first of all, generate our own data. In fact, there was a lot of controversy in the Genome India, and I am partially responsible for building that controversy. I was pissed when I heard that the Genome India data is not going to be shared openly. And you know that. So I wrote an article about it. I write a lot of general science op-eds. And I said, unacceptable. We, the Indian community, use the data generated outside of India so freely. But when it comes to our own data, we are not giving it to anyone. They said data is available only VCS format. I said, nonsense, not acceptable. Make sure the raw data is available with the phenotype data. You can sign a piece of paper. Getting data is how to do it is very well known. So I'll stop here. You will make sure that whatever you generate is in public domain. We must build our own data. We must share responsibility in the globe to maintain the data elsewhere as well. Thank you.
Are there any pressing questions? Okay, then we'll keep that for the later. Just want to say that there are 2 database initiatives, one by DBT and another by ICMR that has been taken up.
I was critical about DBT only.
Okay, so
I can talk more.
yes. So next, Varun, you are here.
But this mic is working, right? Or I can just stand here and give it. So thank you. I think I am a little too young to talk about the grand challenges. However, I have some very, very strong opinion. So, which is all right, right? We all have Bayesian priors. My number one strongest opinion is that our entire field is going to see a major crisis if we do not deliver directly for the government. And we have to deliver directly through therapeutic advances. There is absolutely no way. Money does not grow on trees. We are a dirt poor country. We need to be delivering something in order to be relevant. So this is how I think about grand challenges in the field. What can be done so that we can use the power of big data algorithms to deliver. And delivering is very, very important. Okay, so as I said, right, like everything in my lab and what I feel even for the field, we need to have success stories in order to remain relevant. So in order to have these success stories, the missing gaps are that why is some form of intervention or drug effective in some but not in others? What is the durability of response? In fact, this is where everyone has put in so much money for personalized medicine, but the number 101 thing is, what is the durability of response? Do I need the same dosage? Do other people need the same dosage? We cannot make a fool of the government for a very, very long period of time, right? At some point, we need to answer this question. And then about how to do this drugs safely and scalably, and then personalization challenges. Is this better for me? Is that better for me? So at some point, we need to deliver. So towards this, I think, again, big data is very important, but just by itself, I think, these are my personal opinion, it does not add value and it disappoints the real stakeholders, practitioners, patients, and most importantly, people who fund us. We need to deliver to them actionable stuff. We do not need data dumps. In order to address this, we need big data with longitudinal data in order to have time points, not just snapshot, time points pre and post, and then develop algorithms to see what is changing and then come up with solutions. Of course, when it comes to therapeutics, again, people are building a lot of stories on single samples. We need one intervention across a lot of patients, across a lot of conditions, and then importantly, how to model the microbiome. Until now, we do not even know how to model it. Is it some K-mers? Is it some taxas? Is it some strains? What is it, some metabolites? How to even model it? These, I feel, are some of the important things. So I think as a complex social network with each node as a bag of genes. So again, we have to address these questions. We have to deliver clinically. Why is certain intervention working or not? We have to address these questions in terms of trials. I discussed some of these things, and these are real world trials. Like if patients with kidney transplants are dependent upon stool transplants to help with their AMR, or for other infections in ICUs, or for colitis, or even for wellness solutions, right? I have a feeling that we will all bank our stool at our healthiest self and then use it to reset our microbes later when we are sick. And for all the Cox postulates, we need to follow certain things. If we want to try it, we need to have banks with data across multiple donors, conditions, longitudinal efficacy, and And then this is the thing that my lab is working on. What changes? So can we model microbiome as a social network? Social network where influencers, which may be probiotics. I learned this word influencer in the afternoon. I just love it. A big influencer can be a stool transplant. A small influencer can be a silly old strain of probiotics. How is it changing the network? How long is the network being changed for? So these are the ways in which we can get to the Cox postulates and effector and causal microbes. And thinking of each node as a bag, as just like a Lego blocks, different colors, they may share something, but as a bag with certain capacity. So to summarize, we need to look at the big picture. We have to deliver. In order to deliver, we need to focus on modeling these things well, and then longitudinal data with patients, and how to get the intervention data on which we can build better big data models. Thank you. Did I do good on time?
Any pressing questions? Okay. So I just want to say there's a program at the government called One Health, in which this overarching initiative of joining all the dots, such as a fully contained ecosystem, is being thought about. Maybe, if you can also say something on that.
Yes, thank you. So actually my ANRF career award is completely on the One Health proposal. So we have AMR. So a lot of this is because, and we cannot, even the rich and the poor, we are coming up with the AMR baselines. So everyone is consuming dairy. Everyone is consuming poultry. So if we do not work together, if we do not look into the microbial health of our animals that we are eating, consuming their products, or for farms, or for the shared environment, then our microbes will also be affected. So even a lot of these things, like microbes are shared, antimicrobial resistance is transmitted across organisms and all. So One Health is this beautiful concept where our health is intricately connected to the health of the ecosystem. So, and this is like a shared responsibility, right? So in fact, I have a wonderful thing for it, like one shared world, one shared world, one common health. So yeah, that is a wonderful model, and maybe we can talk more about it in the.
Sure.
Thank you.
Sakit.
A good series of talks that you've organized. So I'll keep it very short. And the central idea is, again, something which you have seen some versions of it during conference. Very simple, my DNA chat, and I'll just give you a pretext of what it is about. So if you think about what AI has democratized, and this word, please remember this word because this is very important for the problem that we want to solve. I can ask questions about, I can use it as a knowledge base. I can ask questions about one particular context. I can ask questions to AI, ask it to generate a hypothesis, and it is able to do a pretty good job at that. And this is a biological hypothesis, by the way. It is very good at generating codes. I can ask it to generate a particular dashboard, which I have no idea about, and it does it very, very well. And then comes the final question about doing variant effect prediction. You saw a version of it in Kishore's talk a couple of days ago. And it works, but it doesn't work, right? So if you have worked with sickle cell or if you're familiar with it, it's a classic example of one single mutation sort of making a haywire change in your entire genome. And this loci is actually the incorrect loci. It happens to be on the same chromosome, but is not the actual location where the mutation happens. And it tries to guess it, and it gets the answer right, but the input is incorrect. So we are very far away from actually having a chart engine which will tell me that, okay, this is my DNA sequence. Tell me what is going to get wrong with my body. Tell me what is going to happen incorrectly as I grow up. And why do I think this is possible is because the main thing, right, and this has been the premise of a lot of talks, and I have some disagreements there that DNA sequencing is all you need, but it is getting democratized, right? So you can sequence your DNA in $100, $200, whether you believe it or not, that still remains kind of a question to be answered. But it is soon going to be possible for all of us to have some version of our genomes sequenced. People have thought about it. So people have already started thinking about how can you go from sequence to tell you about what is really going to go wrong. Like what, if you have a particular mutation, what will be the effect of it? And there is like bunch of work out there. Two of these work are on the protein coding mutations. The remaining two works are on non-coding mutations. However, there's one key area which none of these approaches solve is they assume that one mutation will do everything uniformly in your body, which is actually not true. Because a lot of these mutations are very context specific. And that one mutation is not sufficient because at the end of the day, what we have in the body is a very fine-tuned radio, which is determined by not just sequences, but a number of regulatory marks that actually get deposited. And while there have been efforts to actually look at this in some contexts, we are not there yet. We are not there yet in order to be able to ingest this amount of information coming from individual cell types. And that's where I believe there is a huge gap to be solved. So what should we be doing, right? What modalities do we need? How should we be thinking about this problem? And I think over the years, this problem hopefully will get solved. We require information from multiple contexts, DNA, RNA, proteins. We also want information about what is really happening for a particular genotype. So we need information at the DNA sequence level and try to then understand based on past data that we have collected as to what do we really know about drugs, which drugs do really work. And this is a very generic question, right? You can ask this in any particular context. And that's where I'll ask you to recall the word democratization. Is a lot of these things are getting democratized. A lot of methods that we develop in the lab, we develop it with this idea that anybody in the world should be able to run it on a standard laptop. So if we are able to do that, if we have access to compute resources, if you have, for example, projects like protein folding at home, where every computer becomes part of this effort, something of this scale, we should be able to think about how can we design personalized therapies, right? Again, a very buzzwordy item, but something which I believe is solvable if we approach it in the right way. Right, so if we have access to this kind of data where we are actually leveraging public data, I'm not saying, if I say this to Varun, he'll say that, so what, right? Like, should you sequence everyone? And you should not, right? You should not sequence everyone, because we have tons of data which is already deposited out there. We have not actually squeezed out every possible information that exists out there, something which Binay was also pointing about. So we start there, we generate insights starting from both bulk and single cell data sets. We should design models that are longitudinally ingestible, which means that if I have DNA sequences or if I have information coming from one single donor across different time points, the model should be able to adapt and use that information going ahead. Something which Varun also pointed out. Okay, I'll stop there. And this is what my vision, just 10 seconds, my vision would be something very much similar to a ChatGPT. I go there, I type in my mutation. This you could code in like 10 seconds using one of the coding engines out there. It should be able to tell me what is a variant. It should be able to tell me what are the impact of these variants. What metabolic disorders will I have? What are my predisposition to different diseases? And what are things to avoid, or what are things I should be doing? Okay. With that in mind, just 2 seconds, and India is a genetic khichdi. So before we actually start thinking about these things, while I agree with Binay Panda that the direction that Genome India is taking is slightly worrying in terms of holding on to the data, but it's actually also a process in the right direction, because it's at least thinking about the diverse country that we are in actually making this data available, hopefully at someday. I'll stop there. Thank you.
Thank you. So we'll take your questions later on. Nitya?
Hi, my series of shorts. So I'm going to talk about challenges in AI/ML models for AMR. We recently started venturing into AMR in our group. So this is some of the challenges that we are facing with respect to prediction. So as we all know, AMR is a global threat. We expect more than, let's say, the statistics from TOI says 50, I'm just reading out from here, 1 crore of lives are going to be lost by 2050 if we don't combat it. Fair enough, there are a lot of measures right now, which start with limiting the usage of antibiotics, vaccinations, and there are lots of tools for drug discovery, of course, with computational tools like molecular docking. Then surveillance measures, obviously looking at maybe the sewage and trying to predict an outbreak, like in case of SARS-CoV-2. Using AI for predicting resistance, this is what I'm going to just focus on. And we just mentioned One Health, which is actually where we are not existing alone, we exist with others as well. So when one species is affected, all of us are affected. So amongst these, I'm going to talk about the challenges that we face when we use AI/ML models for predicting AMR. So the first and foremost is we lack sufficient as well as balanced data sets while we try to predict even one antibiotic, resistance towards one antibiotic model. Now the keyword is also balanced, right? So even if we have some, let's say 1,000 samples, if it is not a balanced distribution, then the model doesn't learn well. So I'm also listing a possible set of solutions which we can discuss further. One strategy could be to use generative models, like diffusion models that could possibly learn the distributions of the data sets. But we don't know what are the validations or risks associated with those generative models. The other thing is most of the data that comes from the sequencer, they're not contiguous, right? So we have loss of information. We also have disintegrated, like certain mutants might not be, it is uniform in the colony. So those lead to some sort of noisy data. So maybe we could go for short, instead of the whole genome prediction, certain k-mers and motifs. I guess a lot of talks were also focusing on these. The challenge in any AI/ML model will be interpretability. And in this case, particularly understanding the genetic and environmental relationships. So one possible measure will be to augment the interpretability with a lot of maybe probabilistic and statistical information theory models. And last but not the least, in my opinion, the generalizability of the models is one of the biggest challenges. What I mean here is, if a particular model is trained on a distribution for a particular cohort or a particular population, it need not be generalizable to any other cohort. It may terribly fail in another setting as well. So that challenge, we could just maybe mitigate it with out-of-distribution data, foundation models. Easier said than done, but that's where the solution possibly exists. But as reiterated by a lot of people like Vinay, we need some repositories that are common amongst all populations, among all settings, and the models developed by one group should be actually accessible to the other group for testing, for generalizability, because this is a threat that we can only solve together. So the other part of AMR is also evolutionary dynamics. Like, why is evolutionary dynamics needed? It is not just important to predict the resistance, it's also important to understand the timing so that we intervene at the right time. Using possibly reinforcement learning models and other evolutionary models, one could model when exactly the mutations that would actually make the organism resistant from susceptible. So this is also going to be helpful in population level, models where you can actually know the outbreak even before the outbreak comes. But there are more challenges in evolutionary dynamics. Largely, we start with the extremely large state space, and of course the complexity of modeling random mutations. And I would also say the genotype to phenotype mismatches, because epigenetics starts playing a critical role here, the DNA methylation patterns. So in short, there is a lot to be done here, but it is a very important and crucial step to understand the dynamics as well. So I'll stop here. We'll further discuss
Thank you.
later.
Are there any pressing questions? Okay, then we'll go with Arijit. Arijit is the next one.
Hello. Okay, so I have chosen a more scientific topic, and how we can understand the protein in motion. And I think there was a question to me earlier, like whether we can, those who don't heard my talk earlier, so I work on generative AI for design new molecule. So one of the question was that can we do it for intrinsically disordered protein? So actually this is one of, we cannot. And I think that is one of the problem no one can do. So no one can, because protein is a dynamic and we cannot tell that, we only work with the static structure. So we all know that AlphaFold has two, and there are a number of other groups, not only AlphaFold, which has solved the 50 years old problem of protein folding. But one of the biggest criticism against AlphaFold is that it cannot tell the protein folding pathway. So the picture that I have shown here is that the folding pathway, and this is basically an energy landscape. And what we know that there are many high energy barriers, as you can see here, and ultimately from a denatured state to folded state, it can go. So no method at the moment can tell. Maybe for a small protein, we use methods like MD simulations, or maybe MD simulations cannot also tell that. Maybe we need to use somewhat rare event simulation method like replica exchange or transition path sampling, or some sort of umbrella sampling for other cases where we know the reaction coordinate and there is a large scale question. But why it is important? It is very important to understand this mechanism to understand the disease, because it's responsible for various diseases like Parkinson to Huntington to prion diseases and many more. And these are also, this protein dynamics is also responsible for various diseases, and only we can effectively design a new molecule once we know the dynamics. And I just gave a small example that we did a few years back. So you're working with a protein called HMBS, and it causes AIP, acute intermittent porphyria. So what happens, this is a heme biosynthetic pathway. So various part of this HMB is added. And we just did some MD simulations by manually adding these rings. So these rings are added with the cofactor, and after that it's breakdown. And this is, what you can see outside is that there are many hydrogen bonds, and these are responsible for various diseases, that is AIP. So what I'm just trying to say that we are not yet there even with MD, and this is also the same case for RNA. As you can see that a ligand can bind at multiple positions, and also you can say that for same RNA, there are multiple positions where various ligands can bind, and this is very important to understand to design new molecules as well. So there are a number of methods which are deep learning based methods are coming up. For example, one look at they take the reactive potential, because there are many problems with the current MD. So I just gave an example of such method like torch MD, because we cannot do the charge transfer. Maybe we can use QM methods or QMMM type of things, but we cannot go for a very longer time scale. So maybe that method can solve one part of the problem, because with the deep learning model, ideally we can go much above the microsecond simulation and can see some of those rare event dynamics, which can help. Maybe quantum computing at some point of time, not maybe after 20 years, can solve this problem. But there are also other methods for the unseen part of it. I mean, generative AI or any AI methods require data. So right now, no method, I mean, I have mentioned many methods except this, which has came up, which try to solve this problem, try to see the protein in motion. But what happens that it requires some amount of data. It's done? Okay, I still have, okay. Maybe, I mean, right now there are no method. So whatever I showed in the previous slide, I mean, it requires some amount of data. I mean, maybe 2 conformations, there are 100 nanosecond data point, or there is a big, some amount of coordinate that people, DShaw and other groups had generated in around 2012 or 15. People usually take those things and then try to see from this latent representation some new some conformations, but they are essentially not new. I mean, the model ultimately delivers some states where whatever it has seen. So my main question is that can the new set of deep learning model with diffusion model or other model, can it extrapolate? By extrapolating, I means that the data that it has not seen, can it find out new conformations? So that's essentially the question I'm trying to ask. Thank you.
Thank you. Any pressing questions? Otherwise, we'll move to Millie. Millie, please come.
Can you hear me? Okay, so I don't have any slides. So I'm thinking how we can move forward. For example, now in Singapore we sequenced 100,000 Singaporeans. Now we will sequence 500,000. But I'm not sure if that is the right way. So how we can understand, and my question and my grand challenge is, how we can understand each mutation or modification in each nucleotide, and what kind of effects that have. So how we can do this? There are 2 ways. The one way is just to keep and take longitudinal data with time and collecting. And for blood samples, that is not a problem. But what about tissue? If someone has a cancer, how we can reconstruct this problem? The problems are somatic mutations and epigenomic changes like methylations, and how we can collect them and how we can analyze. So the one approach is we have some cell lines and we intentionally perturbate specific nucleotides and see the changes. Another one is if we have a power to reconstruct, for example, the cancer genome. There are so many somatic mutations. But at the same time, we have many clones of the cancer. And hypothesis is, if we have many clones, maybe we can see development of the cancer. And then, when we have normal cells, and on the other side, we have the cancer cells, and some clones in between, can we use diffusion models in a way how we can reconstruct images to answer this question? Another example are clonal hematopoiesis. So it is known the people above 50, I'm among them. In our blood, there are new clones on stem cells. They are different than normal ones. And with time, they accumulate another changes. And one day that can lead to some illness or maybe cancer, but no. But with that way, if we can collect the data, and again, we have longitudinal information, can we use some methods to understand? So my challenge is how to understand change in each nucleotide. And I think it's time to try to perturbate to understand, not just collect all of information as possible. Thank you.
Thank you. Any pressing questions? Okay, thank you. Yes. Paul?
All right, yeah, thanks. So, yeah, when I was thinking about this, I was thinking a long time, and then I remembered, why am I traveling here? I got a grant with Chirag Jain, and the grant was about democratization of genome assembly. And that's actually one of the grand challenges. So I was like, okay, actually, I've thought of this already with Chirag. But I added the "and analysis" part just to make it go for 5 minutes, a bit long. The idea is very simple. So you have this paper, we've talked about it, the T2T assembly, telomere-to-telomere assembly of the human genome. It's obviously a great achievement, but this is the work of a large group where, you know, money was not a major limitation. They had they had support from Nanopore and so on to provide data. This was like one of these projects that was at the forefront, and you know, we're going to make this happen, and we're going to get the resources that we need to make this happen. So money was not a major limitation, meaning you can get the best data that you can think of. For the assembly process, I don't know actually. I couldn't find numbers in the paper of how long it took, but it was not an algorithm that was optimized for speed at that point. This was first attempt to do something like this. It required assembly expert intervention. So, you know, Sergey Nork and others, they looked at the graph, the genome graphs by hand, by eye, and then tweaked parameters and so on. So that's another thing there. And finally, they had extensive compute infrastructure also. It wasn't an issue. So great, you know, we got this thing. But now if someone in a smaller lab tries to do something similar, they're going to basically not be able to do it. Sequence budgets are limited, and you know, not just like people were saying here in India, but you know, in a small biology lab in the US, you also have a limited budget. So as a result, you maybe are not getting the best kind of data that you could get. If you're a lab that's focused on a biological question, you probably don't have someone who's an assembly expert working in your lab. So you can't rely on that kind of thing to make it happen. You need a tool that's more automated, that doesn't require tuning all these hyperparameters like Mila was talking about. And the compute infrastructure is also usually limited in practice. So you know, what is needed to go from that great result, which gets a lot of attention, now to let's work on making it scalable, both in time and memory, automation. I mentioned automatic parameter selection earlier, and I haven't mentioned this, but in general, better user experience. So a tool that's more friendly for others to use. So I think that's a big challenge after this kind of big result. These are the next steps to make this kind of analysis accessible across the world. I mentioned this already in my talk, this Logan search for sequencing of read archive. I mean, that's an example of, okay, can we make accessible, can we make accessible all this data that we have in the sequence read archive for others to query? And that's, I think, especially important from a democratization point of view, because that data, it kind of has a lot of hidden gems, and you might be able to pull out a lot of stuff, and it's free in the sense of you don't need to generate new data. If you're a big shot lab trying to write a big shot paper, you're almost like tempted to not use this. You know, you want to generate your own hot new data set that no one else has. But outside of that, if you're actually trying to do basic science and with limited resources, it becomes really important for having access to query this kind of information. And so Logan is a step in that direction that people have done, but a lot more needs to be done there. And there's a lot of research opportunities here. Thank you.
Thank you. So now we move on to the next part of this presentation, and we will have now the entire floor open for questions from the audience, and would request all your questions to be precise and not be, voluminous and meandering, and be brief. Okay, so we'll have the chairs on the podium and request all the panelists to join us on the podium. So we have heard all the presentations, and I'm sure there are questions that are bubbling in your head, which you can ask the panelists directly. To start with, Varun asked me to tell something on longitudinal data. So some of you may know that ICMR and DBT has taken initiative to create databases, data sets. Of that, the ICMR initiative is actually anchored in IAC. And I am the PI who is taking this forward on behalf of ICMR. It's a national program. However, getting longitudinal data onto the database is a regulatory nightmare due to privacy concerns. So we still do not have all those algorithmic techniques, especially blockchain kind of a technology, deployed on scale, such that we can ensure that all the data that we gather for patients, and especially when it becomes a longitudinal data, which means if it gets exposed, then the entire history of a patient will get exposed. So some of these regulatory hurdles are steep. And in India, the technology stack is being built, but it is still not there. I'm not sure what is the case with the United States. Any idea?
This to me?
Yes?
I don't know.
I don't know. Okay. Anything about Singapore? Okay.
I know that we need to take care about privacy. So, for example, when we analyze this data from 100,000 Singaporeans, so we should either use AWS because they have all protection of privacy, or we should switch off internet and analyze data. So it is not easy, but it is important to keep. And there's some new legislation that will be probably soon available about that. But also there's the option of using federated learning, so that you don't have the direct access to data, so and you still can learn models. So I think that there are many initiatives in this direction.
So,
Can I add something to this?
yes, please.
This is actually where I think India has a brilliant opportunity. If we summarize a lot of topics that everyone raised, we all talked about databases, we all talked about interventional data, longitudinal data. I think with some privacy protection, if we can get the data of, let's say, we talked about tumor evolution, right? The clonal evolution in different tumors after different therapies. I think if we can somehow get some kind of, if we can biobank the samples through a national registry, and of course we can decide on a few interventions beforehand, like biobank the samples for the technologies of future, and get some kind of metadata, or at least collect it and put it in a vault. So tomorrow, if there is a better technology, better approach, we can somehow reuse this data. I believe that is the key. So somehow if we can get together people, stakeholders, at least biobank, let's collect the metadata, put it in a vault. Do not give access to anyone right now. But let's not just throw away this sample forever.
So some of these, initiatives of whatever you're saying, and they are present in institutes like AIMS, where they are biobanking, but in a limited way. Yeah. But to actually deal with problems of scale that we face in India, we need, because of diversity, actually we need solutions to be deployed at scale. And for that, we need technology. Because otherwise, AIMS is one I know. There are several, the Tata Memorial has biobanks, I know. So there are several other hospitals which actually do these things.
So I have a point. It's a little radical idea, and I like it. You must see what are the best use cases around the world. Genome in a bottle. Who makes it? Justin Zook. Who is he? He's a scientist who works in NIST, that stands for National Institute of Standard and Technology. You must make data de-linked from investigator once publication is done. That's the only way to make data available in perpetuity to everyone. You must not keep the data in charge to investigator who generated it. They hatch like they're hatching the egg. They never give the data. Once the publication is done, the data, whether it's longitudinal data, whether it's non-longitudinal data, data must go out of that institution, be housed in a central repository who are not doing research based on that data. NIST in the US is an organization which has made standards like ISI in India. That is what we need to do. You make scientists who generate the data in charge of the data, you're screwed. You'll never get the data. Once the publication is done, the data and the information, metadata, has to go out of that institution, be stored in a neutral organization where there are 9 to 5 job holders. My job, my salary comes if I just maintain the data. I'm not asking scientific, so I have no conflict. But if I am the investigator who generated the data, why will I give? I have 5 more graduate students in the line. I will publish the paper. We are all familiar with it. Come on, let's be honest. So the question is, you must de-link data from investigator. That is the need of the time. Therefore, the IBDC was formed. I was actually one of the drafter of the biological data sharing guidelines in India. I know we have a long way to go, but that's the right thing to do. This decentralization of data and information. Investigators get money, get grant, generate data, publish papers, but send the data out to a central repository where the investigator is no longer, why should I ask an investigator for data? I should ask the data bank for data. I ask NCBI, I fill the control access form with DBGAP. I don't ask control access data from what Paul has generated. Once it's published, it's done. This needs to be done. Unless that happens, you will never get data.
Saket, do you want to add on, like you were talking about individual tissues and all? So how can we biobank samples to get to that?
Yeah, I think just to follow on to Vinay's point, right? So I think DBGAP model is very successful actually in the US, where once you deposit, so most of the research, as long as it is happening in India and it is publicly funded, I think it is actually the moral responsibility of the person, because it's public money, to deposit that data into some public resource, right? It's very simple, not very hard to understand. There are legal implications, of course, if the data goes out, if there is privacy and so on. But there are also other mechanisms which, you know, we can think about how to sort of streamline it. Unfortunately, India doesn't have an equivalent of DB-GAP yet, but I think we are actually marching towards that direction. I think that's the right way to go towards. Coming on to the biobank, I think the biobank are also facing similar issues where the hospitals right now, we in India, we have a trust problem, right? Which is, if I give this data to this person, then he's just going to run away with it, publish 5 papers, 10 papers, and not make us co-authors, or we'll be treated as secondary people. And, you know, like all those issues, I'm sure we all in this room understand what is really going on. And I think that requires some efforts to solve that problem, which is the hospitals. For example, Varun mentioned Tata Memorial, and I sit in that same city where Tata Memorial sits, and I see there is a friction, right? So if I have to go multiple visits, still I have not had access to data. And again, I'm not blaming anyone. It's just that trust problem which we have to solve first. And there are ways to do it, which is to sort of have all the stakeholders on the same line of thought, that nobody is working for anyone. We are all on the same page. We all want to push things forward. We all want to make science forward. And that requires everybody's effort, right? It's not my job, it's your job as well. And we are, as long as we are moving in the same direction, I think we should be able to solve some of it. Unfortunately, at least so far, I've not been able to solve it, but I try every day to sort of bring in that trust somehow. That even if you're not just bringing in data, but you're actually trying to, we are working towards the same aim, right? So that's the motto that at least I try to work with.
Before that, do you want to add something on AMR, like on the One Health, how they are successfully sharing the data? If that template help us, perhaps we will be more familiar with it.
I had a point about the hospital data as well. So in India, I feel a lot of data has been generated. I mean, it is being generated every day in many hospitals. It could be related to AMR, it could be related to cardiovascular emergencies. So the issue is people just do not maintain it. And because of that, those vital data is lost to the research community. For example, antibiotics have just been given just left, right, and center in so many hospitals for so many surgeries. And if they are just documented, monitored, and in a central place throughout the country, it would, I believe, help quite a lot. So I think that's sort of a central system in which one could, maintain hospital data, or they are forced to, whether they are private hospitals or government-run hospitals, if there's a mandate to monitor the antibiotic usage in all the hospitals and provide it to the government, which I mean the database, I think the research would develop quite a lot from that.
Okay, if you want to go.
Yeah. So I thought, there were a lot of comments about data sharing, which I agree with, but there were specifically comments about Genome India. And as someone who's been part of that, I thought I'll just add a couple of clarifications there. So one thing is, so I do understand that the amount of time it was taken to put together this was much more than what we anticipated. But everything that was discussed so far, which is that this kind of data should be out there, it should be shared at a central repository, and it should have proper access mechanisms. All of these have been thought of. In fact, all the raw data and the processed data and the phenotype data is deposited at IBDC. And there was a call for proposals, which I'm sure you've read about it. And I am told that there are also a lot of applications for it, which means that data access will be provided soon. But the reason why we took so long is because we wanted to ensure that we did not put something wrong out there. This was a project that was happening at this scale for the first time in the country, and there were a lot of learnings even for us. There were many things that did not come out as we expected. And there was a lot of brainstorming in actually making sure that what we are saying, we can say with some confidence. And that took much longer. In fact, I know that even for phenotype data, It's only a fraction that we could still put out as of now because the data needed so much of cleaning and it is still going on. So all this took time, but now the data is there. And as for access to raw data, I don't think the question ever was whether raw data will be accessible. It was, I think, it was at best I can say it was probably a lapse in communication, because what DBT meant is that there would be no data download, fast queue download. And that is actually in line with other global consortia who do not give you a copy of data to download. Okay.
Wrong.
I must say, okay, two things. This is not about investigators. One thing absolutely clear. DBT, there was no miscommunication. It was black and white. VCA file will be given. What is
For download.
There was no word download.
Yes.
You can access
Which is
it. You can read what you want. I'm reading what is there. Raw data will not be available. VCA files will be available. That is a screw up. They have solved that now. Because of it is, hold up, you are with us. You are a part of the community. We are talking about so this is the distinction you must understand. It is not about an investigator against another investigator.
But I'm not taking sides here. I'm
I'm
not saying that I'm on their side.
You appeared giving a clarification. You shouldn't. The reason is that we are on the same side. The reason I'm saying this is investigators generate data. Once investigators generate data,
Yeah, but
it's
it
out.
is already delinked. That's what.
That is what I'm saying. So that means you are out. As soon as you deposited the data in the database, you are out of the question.
Yeah, but I had to justify why it took time, because it
That
needed
was not the
cleaning.
question.
Yeah, and the second thing is, please tell me which consortia like UK Biobank or Genomics England, which of them
I
actually
have downloaded zillions of data from GDC FastQ file. I will give you access if you are in one of my collaborators. What are you talking about?
I think I can just add something. So NIH is now moving towards a cloud-based model. So I agree there, like where they are sort of making you do everything on cloud. But it took them a while, and I think it works there because people have access. You can use your funding to buy.
Sorry to interrupt, Saket. The reason that the federated structure is going to be in place because the size of the data is much easier for you to move compute rather than move data. Right. It has nothing to do with access. Don't get confused. Because the data amount is so much, you cannot download fast data. So you move your compute to their cluster. So federation helps. So it is not because there was a mandate against downloading fast queue data.
No, I never said there is a mandate, and
no,
it's also not.
You mentioned, it appeared like you mentioned it as for the global standard. The global standard has come. I'm a part of G4GH. I'm a part. Look, the global standard is that there are mechanisms to share data, control access data. You as an investigator, your institution as your parent institution has to file the form, has to do this. The same mechanism can be involved. I think we have to be dirt honest in our country. Investigators do not share data. That includes me. Now the question is that we have to get away from that mentality. Enable our students, enable our Indian community, and if I may say, enable the community which is elsewhere. We have been the beneficiary of GDC in the US data. Why should the American scientists not get access to Indian data? The American taxpayers has paid for the SRA maintenance. You all sitting in this room, at least the investigators have been the beneficiary. Why should my data, Indian data, not be accessible as long as there are certain restrictions which are fulfilled? That's the point. And I think what I have heard recently, and I had in communication with the funding agency, they are moving in that direction after the hiccups. I wish the hiccup wouldn't have happened if they have consulted us. They did not. They unilaterally did it. But now things are moving in the right direction. Let's hope that things get resolved.
Yeah, I think some of the things do take time in India, and with hiccups, things settle down. So we hope things will be in place, and we'll try to move the discussion toward more scientific oriented issues and discussions. So you have scientific questions, right?
A lot of metadata, like a lot of these biological samples, wouldn't be completely useful if we have the phenotypic, like the metadata, and the clinical aspects to it. Out of which we do find a serious problem because a lot of rural hospitals, they don't collect in an electronic database. We see it in big hospitals. So how are we going to incentivize the full use of these biological samples if we are not going to have the full, I mean, we don't need the names and all, but we do need the patient history and all. So how are we going to do that?
I have one small thought on this. Maybe we can come up with a credit system. The data that if you are giving this information, then you can also access the credits back for downloading it, because like I firmly believe that there is nothing called goodwill and good karma. It's all about incentives and a deal. So somehow giving them credits for giving this information so they can get the credits back if they want something else. This is a feasible model in my mind. Maybe other people can
Yeah,
add to
so
it.
I think you have to, there's a legal, setup here in India where the Supreme Court says that the data belongs to the patient. He is the owner of the data. So if he or she is fine with the data being donated, as he suggested, and everything is fine. But to be able to, operate all these modalities, the infrastructure needs to be in place. Right now, if you get a sign in a paper and that paper gets lost, then you are in trouble. So many of the hospitals would not want to engage in such things because they are basically beckoning trouble. But there are many well-meaning hospitals which actually want to contribute. But what we are lacking are, as I said, technology solutions that will be able to deploy these concepts and make it, scalable and deployable all over the country. So I think there is a movement, but as you can understand, we are a huge country with a lot of population and a lot of diversity.
Rural communities. Again, if it is a big hospital, we have the electronic setup. Like we have everything in a file, like which could be, I'm not saying accessible, I'm saying keep properly maintained. But in rural hospitals, it's not properly maintained. So at least if we start maintaining something, then we could.
Digitization process being enrolled out through the ABDM. You may have heard of the Ayushman Bharat. Some tech stack is being built. There's a national health authority which is spearheading all these initiatives. So the hope is that these digital infrastructure would be in place in times to come. Okay, so there is some movement in this.
I'll just add. So I think this is a common understanding that if it is rural, there are no systems in place, but actually that's not true. And the easiest way for you to find out is you just search something called HMIS, which is the Hospital Management Information System, which puts out a lot of public data coming from variety of settings, which actually includes the rural population. And they are actually monitoring a lot of things, right? It might be not at the patient level, but very simple metrics about how many people are being born, how many people are dying. That's all actually being recorded very, very well, right? So India often, whenever you come across India, like you'll think about, okay, we're not keeping tally of all this. Actually, there are actually systems in place which are doing this.
So what is lacking in India is interoperability among systems to talk to each other because there is no standardization. There has not been a policy guideline that has been applied so that different databases can talk to each other, they can interoperate. You can pull in data from various places and then get going with your analysis. So those are the challenges that are being worked out and can only be worked out once the technology is in place. Because then you have to pull in the retrospective data, make sure that they fit into the framework that you have built. So it's cannot happen overnight because we haven't gotten started very early on. Yeah. Other questions?
So to continue madam's question, the thing is, being a clinician, suppose at the primary care physicians, first of all, physicians may have to be the method of collecting the data, because just data should not become a garbage like thing, because being a physician we don't have much of the technology knowledge, except in medical clinical applications, or in the institution like research institution like NIMHANS or anything, they might have exposed to give up this data for the specific individuals, for the specific organization. The method of collection of the raw data from the patient as such. Of course, sir has told we have to go from the patient's undertaking and all these things. Apart from that, is there any specific methodology for applying these data banks method of collecting the data exclusively from the primary care physicians, including the rural or corporate or government institution, medical college institution. Any specific methodologies are available for the physicians? Standardized methodology, what I mean to say. Forget about the demographic data and other things, nationalized data. But if I want to become an investigator for specific like multiple sclerosis or Alzheimer's disease, for data banks supposed to give it, is there any specific methodologies are there?
Maybe I'll have to answer that question. So there is something called a case record form for every patient that gets admitted to any health center. So that's a case record form for an interoperable entry into a database has to be put in place, which then should get encoded through a standardized dictionary that then can be taken to any place and then used for analysis. So the point is there are not one company, there are many companies which have EHRs, right? So every company has their own CRFs. But then ABDM, which is our national initiative, it is also creating its own. And the hope is that the private hospitals would have a middleware which can actually then convert that data which is collected in every hospital into the ABDM compliant format so that then now it can be shared all over. So some of these things are in offering.
Can I just add something to this? So I have thought about this a lot, discussed with a lot of people. The solutions are all over the place, and it is absolutely bonkers to expect the clinicians who are already overburdened to collect this data. It is dead on arrival. The only way this can happen is, and again, we are a democratic country, and that too, perhaps a little much too much democratic. We have made all these privacy laws aping the West and all. The only way is to monetize the data. As soon as you can monetize the data, patients can sell their data, companies will rush to get this thing. Just recently, I heard that there was an open call for 5,000 lupus samples. Can we get lupus samples before and after? Right now, there is a lot of money riding on it, but the regulatory part is not clear how to get the consent for the patient to sell their data. So this is the only feasible model in which there is some sort of monetization that is a part of there where the hospitals, the physician and the patient all get to own, all get to some monetary benefit. That's the only way in which we can come up with scalable, easy solutions to harvest this data for science and for greater good. But we have to include some sort of monetary component. This is my understanding.
You may call me socialist, but I disagree with that.
We'll keep away from non-scientific questions and we'll stick to scientific questions. Okay.
I would like to know, what are the technical difficulties? When I heard everyone, mostly the problem was data is not available, data is not there, or we are not able to share it. Most of it is it looks like there are some technical difficulties, in building the platforms or in building the correct guidelines. But coming from an institute, they are doing things like these on daily basis. Like simply creating a database that is secure enough for data that have some problem like legal problems. So there are certain frameworks that can be applied. So what are the technical difficulties researchers on other end faces? So people who build these tools and database we can work on.
Any of the panelists want to answer it?
So the question is, what are the technical difficulties for data sharing?
Yes, I think that's in a nutshell.
What are the defects other people have been facing?
They have been doing it for decades. We need consensus at the government level, different ministries. We need to come up with policies. We need to come up with guidelines. It's nothing new technology. This technology is old.
The framework has to be in place where this can be enabled.
Yeah.
That is missing. Okay.
I think confidentiality is one issue, but I think there are a lot of ways to deal with that. And as he mentioned a few minutes back, that there are things like federated learning, blockchain, and I think there are lots of companies even in India which are offering these solutions. So I think we should
try to take those help, because confidentiality is one issue. Because these data, if someone know the genomic data, that will be difficult for future.
Uh, yeah, it's not just scientific, it's a bit of technological question also related to some policy aspects, like, so I wanted to ask this to Professor Binoy, but everyone please feel to answer. So we have been talking about, making data open, but let's say in a country like ours, what do you think is the best model to fund it? Like, who actually funds it? Is it the state, given the capacity it has? Or like in the US, I've come across certain organizations who are doing some, democratic autonomous organizations where they have this blockchain kind of system where there is a community that is responsible for funding and also has credits to it. So what do you think, what kind of model would work for our country, and would such technologies like blockchain be useful in those efforts? Thanks.
So I think the best organization to fund is public exchequer. Public exchequer funds it in perpetuity. That means your taxpayer money goes in funding something which is for all Indians. Whether you are working in an academic public institution, academic private institution, a company, everybody is Indian. But I think the responsibility of building different components and maintaining it till perpetuity falls with public exchequer. That's the only way the government has to do it, house it, put guidelines, put policies, put mechanisms of access, then anyone can access it. Because you can't give that responsibility to a private organization. I don't think that's their job. It has to be public, but of course, private organizations are also Indians. They should also be able to access the data. The guidelines should be there how to access the data. That could be, there could be difference in different organizations, individuals to access. But I think how to make it, who makes it, who maintains it, must fall in the hands of public exchequer.
Apart from this, my question is with Professor Paul, that what major difference would we have while working with unitics and contigs versus contigs?
What is the difference between unitics and contigs?
Yeah, like which will be more significant, or which will be more which will provide good results during the assembly, if we work with unitics or if we work with contigs?
So you can view all unitics and contigs as both kind of a downstream of the raw data. So unitics are more faithful to the raw data. If you say, I've got my reads, I'm going to build, so one common thing is, I got my reads, I get all the k-mers out of the reads. Now, k-mers that are singletons, they appear only once, I throw them away because they're probably errors. The rest of the k-mers, I put them in the graph and now I create unitics. Those unitics represent those k-mers exactly. I didn't do any post-processing. To get to contigs, people do now kind of processing of the unitics to do more error correction, so more aggressively remove things that they think are errors. And two, often to simplify the graph, it's often removing heterozygosity from the graph, depending how you do it. And from there you get contigs. So contigs are more processed. If you're an assemblers usually will output what they call contigs because they need to do those extra processing. When I want to represent a set faithfully, then I stick with unitics. Or if I want to kind of, assembler outputs contigs, but I want something kind of more raw than that, maybe for the AI, whatever, I don't want to, I want to keep as much of the data as possible, then I might use unitics.
Thank you, sir.
So you were talking about, Paul, you were talking about democratizing genome assembly. So I think the estimate was right now it takes about 2 weeks. I think one aspect of democratizing would be to make it faster. To me it seems like, and please correct me if I'm saying anything wrong, to To me, it seems like it has to come close to a resequencing-based assembly in terms of runtime for it to become really effective and for people, large number of people to use it for various different things. Which means it sounds like it has to have more than an order of magnitude speed up, probably 2 orders, which will not which will require not only algorithmic improvements but also probably HPC kind of stuff like using multiple threads or multiple nodes and what. So does this sound correct? Firstly, and secondly, what is the current state of affairs and what all needs to be done?
Yeah, that's a good point. I think, and I think the interleaving, the mixing of the algorithmic improvements versus the HPC improvements, that's an interesting question. I'll give an example, going back to short read assembly. K-mer counting and unitic construction were, in first assemblers, they were very slow. It was similarly, took a long time. And there were approaches that were using probably even GPUs, but for sure multi-node and HPC type stuff to build fast K-mer counters and to build fast unitic compact. Like there were several papers how to do unitic compaction fast. And I think they turned out in retrospect to be premature because there were still a lot of things you could do algorithmically and just being like more cache optimal in terms of how your algorithm runs that, and just exploiting just basic threads that once those people figured out those algorithmic improvements, it made the HPC approaches based on previous algorithms, it dated them, right? But I think I think maybe what I'm getting at is maybe the algorithmic improvements have to be first and kind of stabilized to a point where like, okay, I don't know how to get another order of magnitude speed up. And that's when we can kick in all the HPC optimizations.
So where are we right now?
For T2T assembly, I think we are still need a lot of algorithmic improvements there. I think I'm guessing, right? But this is my best guess, is taking those current algorithms now and trying to, making them on HPC, speeding them up. It will be a little premature because then the algorithms will improve, and then you'll need new HPC versions of those algorithms.
Thank you.
So we're almost out of time. So short question, no?
My question is, how far have we landed in developing any computational tools to actually classify the genomic signatures and assign them to the tumor topography and get some tumor heterogeneity scores that would actually be beneficial for the patients and to develop some sort of therapeutic response?
What do you mean by we?
So anyone who is like confident,
But
like
what do you mean? We means, do you mean individual researchers, or what do you mean by we, or as a country? I mean, I didn't get the question.
As a country as a whole. So what advancements have we made in that respect?
So are you asking, if I paraphrase your question, are you asking how far have we come in cancer genomics?
Because yes, cancer genomics, and specifically pertaining to the population, Indian population.
Yeah. So there have been several small efforts because our grant sizes are small. So I can tell you in the last 10-ish year, there have been several efforts. The efforts like, for example, which is a part of the International Cancer Genome Consortium efforts, which was held in the National Institute of Biomedical Genomics for oral tumors. There have been also other discrete efforts on breast cancers for oral tongue tumors by our own group, and then also colon cancer, gallbladder cancer. So there are multiple groups individually and in collaboration with clinical researchers have published, deposited many individual papers, but each of those have limitation in terms of the sample size. None of those are more than, I believe, 150-ish is probably the largest I am aware of in the whole genome cancer. Now, because of the size of the grant is small. So that is the reason why Genome India, as Tej was saying, this is important because this for the first time had 17 different institutions coming together to do sequencing of, obviously these are asymptomatic normal individuals, not cancer. But in cancer, these studies were small, it's all published, it's there, and people are continuing. And if you really look at the global efforts there, TCGA leads the effort because there are more than 6,500 samples there, each with nearly 600 to 800, depending on which cancer type you're talking about, and people have been using extensively. So if you really want to go one step further, then you will have to come up with large grant size, multi-institutional grant size, where you can take like maybe 5,000, 10,000 samples. But you keep in mind, there's an interesting, beautiful paper by, I believe, from Broad Institute in 2013, where I'm forgetting it's from Mike's group. I'm forgetting the first author, who I know of well. Uh, in one axis, you have the frequency of mutations. The other axis is how many sample pairs you need to actually do. So for most of the tumors, it comes out to be, if you want to go down to 1 to 2% frequency, you need to actually type 10,000 or more samples. You getting my point? So we are very underpowered. But if you're talking about 5% or larger mutation frequency for the large big guys, then obviously you are okay with 500. So we have a long to go, but there have been a lot of efforts on the ground.
May I answer and maybe connect the last two questions? So when they started with telomere to telomere consortium, so there have been many assemblers and none of them were optimal. And people decided and they put together efforts and they spent a lot of time to construct the first one. So it was CHM13, it was 2022 or something like that. And next year they sequenced and using the same approach, they assembled chromosome Y that missed. And at that time, they needed lots of ultra-long data, hi-fi data, any kind of data. And I was involved and I decided, okay, is it possible that we use just ultra-long reads so that we don't need to all other kind of data? And we suggested correction and it worked. So at that moment, 1 year later, you have opportunity that use only ultra-long data to reconstruct the genome. Year after us came Hao-Yu and Heng, and now you can use just ordinary nanopore data to have decent telomere-to-telomere genomes. So that's the way how the things can move forward. So it's the same way in the cancer. Now we have the cancer genome in the bottle led by Justin Zug. I'm also part of this consortium. So soon we're gonna have the first cancer genome reconstruct. Maybe not perfectly, but it will be. And then this is a chance for the improvement in algorithms in all of this. And it is not only algorithms for de novo assembling. Now I think that one of the biggest obstacles are base calling. Base calling may be more expensive than actually algorithms for de novo assembling because they need GPUs and lots of power. So the way is just sequence one genome correctly and then the progress will come. So if we try to solve all kinds of problems, there is no way to do it.
Thank you, Milay. So we have run out of time. Should we ice the time for something more or we should end? Okay. Yeah, so I guess we have covered most of the questions that we had from the audiences. So let us put our hands together for the panelists. Thank you so much, and we appreciate your comments, and we hope that it was of use for all of us. Over to Chira.
Thanks. So I request all panelists to go back. So thank you.